Dr. Hurd,
Yes.
I finished my exam today, but it did not. Did you know that it's not grading it?
It probably doesn't. I probably don't start grading it.
Oh, you grade it or the computer?
Well, it somewhat grades it automatically, but I also kind of go through. So that's probably why you don't have it, because I didn't expect anybody to finish their exam already. You're a little bit ahead of the game here.
I know.
You're already done with the class.
That's what you get when you're at home doing nothing on
nothing
bed
but recovering, right?
Yeah.
So now you just intimidated all your classmates that have joined it.
I was trying to say it really quickly.
But I'll take a look at it tonight and let you know. Okay.
And then I'm gonna submit my extra credit, because I do got my extra credit too.
Okay.
Why not?
Why not? I don't think you need it, but okay. All right. Okay, now Kiara's just intimidated everybody else in the class.
No.
All right. So we're going to start going through the endocrine system. So much has happened since we last saw each other just a couple of days ago, you guys. Can you believe we were just talking about Omicron and how I said, and I apologize, I keep getting messages, my internet's unstable. The Omicron, I just watched Dr. Fauci tonight. Now they have a case of it in San Francisco.
Oh, great.
Yeah, so it's here on our shores. And then crazy, another school shooting, if you can believe that, here in Michigan. I'm sure you all heard that news. And, it's just like, what a joyous holiday season it already is, right? Between virus and school shooting. So, that one of the questions at introductory health meetings now, you've probably had it in your own life with your own physician, is asking people whether they, own guns. A lot of people are offended by that, but it's the same thing as asking people whether they consistently use seat belts. You're just trying to assess for people's overall health and safety, and if they're practicing behaviors that save their lives or put their lives at risk. So somehow I have this thing going across the bottom of the screen here. Zoom seems to have its own mind lately, just like Blackboard when they updated it, that it's now transcribing everything that we all say. So I have to figure out how to turn this off, I guess. All right, so endocrine system. We're moving on from nervous system, but we're still working with cell-to-cell communication. So we'll see the big difference between the way the endocrine system communicates cellularly throughout the body versus the synaptic transmission at the nervous system. And they use a lot of the same chemicals. When we use them in the hormones, when we use them in the endocrine system, we'll call, for instance, norepinephrine a hormone, but of course it can be also a neurotransmitter when it's at the synapse. We'll differentiate between the different methods of action of steroid versus non-steroid hormones, and we'll look at what are called the endocrine reflexes of upregulation and downregulation. And then we'll look at these funky chemicals called eicosanoids, which is a class that includes prostaglandins, thromboxanes, and leukotrienes. And then we'll talk about hyperactive glandular secretions and hypoactive or low glandular secretions. All right, so both our neuro system and our endocrine system maintain our stable environment of the internal systems of our body. And they do this by allowing our cells to communicate with each other. and to integrate all that information so that we can exert ultimately some level of control to maintain that consistent internal environment. The neuroendocrine system, putting the nervous system together with the endocrine system, function through all these chemical messengers, whether they're neurotransmitters or hormones. And as I said, sometimes they're the exact same chemical. Just when they're secreted from a nerve to act on a postsynaptic nerve, it's called the nervous system and a neurotransmitter, and a hormone is secreted directly into the circulatory system. All right. In general, neurotransmitters act rapidly and they're short-lived. They have to just travel across that little gulf called the synapse. Okay. And these will control only the muscles or the glands that are innervated by the nerve fibers. Whereas in the endocrine system, hormones last longer and they travel farther. They're secreted out of the gland directly into the blood. And from the blood, they travel through the whole circulatory system throughout the entire body. And so they act on many, many, many different cells. And they can regulate most body cells as long as there's a receptor for the hormone. So we call our endocrine glands ductless glands compared to our exocrine glands, which we've already talked about, like our sweat glands that secrete sweat through a duct onto the surface of epithelium, or like sebum, a secretion that is also secreted out of a duct onto the surface of the epithelium. The glands of the endocrine system are widely scattered throughout the body, and they have a multitude of actions. And the endocrine cells will be manufacturing these hormones and secrete them directly into the blood. There's a few endocrine glands that are made out of what's called neurosecretory tissue, which is kind of a modified neural tissue that still secretes these chemical messengers into the bloodstream, though rather than across the synapse. And the example, a good example of that is norepinephrine, a hormone when it's moving through the blood, but a neurotransmitter when it moves from a nerve to a postsynaptic nerve. All right, so here's to remind you, you have your endocrine cell. Of course, it's going to have its endoplasmic reticulum that will manufacture, if it's a protein hormone, for instance, and pack it in a secretory vesicle and secrete it outside of the blood, outside of the cell, directly into the blood. And it's going to take some time to travel through the circulatory system to get to a very possibly distal, far away site on a target cell. We call it target cells, and it affects target tissues and target organs. In the nervous system, the neurotransmitters are made up in the cell, and they're packaged down also in secretory vesicles. And the action potential causes the rapid discharge just across that small space of the synaptic cleft. So the effects are localized rather than generalized throughout the body, such as a hormone. And here's a very busy slide. This goes through all of our major endocrine glands. And so let's kind of walk through it, going pretty much from top to bottom. So up in our brain here, the hypothalamus, we've seen it before. Remember, the thalamus is the big egg, the hypothalamus, hypo below, just hangs from the thalamus. And this is sort of the master gland of our whole glandular system. And it will secrete a number of different hormones that are called either stimulating, releasing hormones, and they will stimulate the glands, other glands in the body, and compel them to make their glandular secretions. So everything, sort of a lot of our basic functions are under hypothalamic control, such as all the secretions that are released from the pituitary gland, things like your water balance, your sleep. We're always talking about the hypothalamic control of our temperature system. as well as appetite and blood pressure. All right, back here, directly back here from the hypothalamus, we have our pineal gland. Remember, this is what secretes melatonin that sets our body clock. And there, hanging below the hypothalamus, is our hypophysis, or the pituitary gland, okay? And the pituitary gland is also known as the master gland. So we have the hypothalamic master gland, which really is the master of the pituitary. And then the pituitary itself, also called the master gland. And I know that's confusing, but you will see both, all right? So I guess think of the hypothalamus as the grand master, okay, because it does control the pituitary. And we know, sorry, kind of crazy slides here. The pituitary sits in that sella turcica, really guarded because of the importance of all of the secretions that are coming from the pituitary. And we'll go through this pretty directly. The pituitary creates a lot of hormones that will stimulate things like the thyroid gland, and it will cause the thyroid, it will release something like thyroid stimulating hormone that will cause the thyroid gland to then release its hormones. All right, we'll go through that a little bit more in detail later. Down below here, we have the thyroid gland, butterfly shaped in your neck, and lying on top of it, some little nests of parathyroid tissue. Okay, and these parathyroid glands, we talked about a little bit before with calcium control with the bone. So you should be familiar with them. So really key role in calcium regulation, and your thyroid gland, of course, controls your main metabolic function. All right. Down here below that, we have the thymus gland, lies in the anterior chest cavity. And it helps to produce the white blood cells. Your thymus gland is really prevalent at birth, and it slowly involutes and loses a little bit of function gradually, every year over your life, till you basically almost have no thymic function when you're elderly. Moving down, we have our adrenal glands. Adrenal, add next to the kidney. So these lie right here at the superior poles of the kidney. And your adrenal glands are going to make your adrenal hormones, epinephrine and norepinephrine, adrenaline and noradrenaline. These are going to help maintain your blood pressure and also work with regulating some of the metabolism. Big gland in the middle of our abdominal cavity, kind of lying a little bit posterior below the stomach, is the pancreas. Okay, we talked before a little bit about exocrine pancreas, but now we're going to get into endocrine pancreas. And endocrine pancreas is going to produce that hormone we all love and know so well, insulin, the lack of which, of course, causes diabetes. All right. Oops. I'm sorry about these slides. This trigger happy mouse I have. All right. And the pancreas also will produce its counter-regulatory hormone of glucagon. So insulin moves sugar out of the blood and into the cells, and glucagon is mobilized when we are low blood sugar, and we'll put more sugar out into the blood. All right. Then we have our reproductive glands. In a female, the ovaries that produces estrogen, and in the male, the testes that will produce the androgens, mainly testosterone, right? Estrogen and progesterone are our main female reproductive hormones. So the hormones are the chemical messengers, and their effects are felt by whatever receptors that they sit on. And we can classify the hormones in different ways. We can call them tropic hormones if they're hormones that stimulate growth and secretion, things like thyroid hormone is tropic. Things like insulin is tropic. Things like especially growth hormone, okay, obviously helps with growth. Sex hormones, we know, are going to target the reproductive tissue: estrogen, progesterone, testosterone. Anabolic hormones are another category related somewhat to tropic hormones in that they stimulate complex molecules in the target cells to be building up portion of our metabolism. All right. And you'll see the anabolic hormones we know very well as being abused often by those who are trying to get a competitive edge in sports. Steroid hormones are all manufactured from the base nucleus chemical of cholesterol. Okay, so they're all going to start out from cholesterol and get modified to some degree. And there's overlap in all these because you will see our sex hormones are largely steroid hormones. Okay. Anabolic hormones, like I said, can be considered tropic hormones. And then we have what are called our non-steroid hormones. These are not made from that nucleus. They don't look like cholesterol. So these tend to be things like proteins, polypeptides, and amino acids. They tend to be water-soluble hormones. All right, so let's first look at the hormones that are amino acid hormones or derived from amino acids. These are the most numerous are protein and peptide hormones, ranging from just like 3 amino acids to over a couple of hundred amino acids. And we'll see insulin is one of the most characterized hormones. And this is made up of 2 polypeptide chains that are connected to each other by disulfide bridges from a sulfur molecule to another sulfur molecule between 2 sulfated amino acids called cysteine. Glycoproteins are going to be a lot of the proteins that are released from the anterior pituitary gland, and they have their protein subunit portion to which sugars are connected. So a glyco portion, the sugar portion, and the protein portion. Amine hormones are small hormones, and they're synthesized by modifying a single molecule of either tyrosine or tryptophan. So tyrosine is an amino acid, and modified, it can yield what are known as our catecholamines, which are epinephrine and norepinephrine. These are secreted, as you know, at the adrenal medulla, those adrenal chromaffin cells. And once they get into the circulation, they're pretty rapidly broken down. Tryptophan, we all know from after just having had our turkey dinners, we release a lot of tryptophan, and this is what helps make us satisfied and tired. All right. Tryptophan will make our thyroid hormones. This is made by combining 2 tryptophan, the conjugation of 2 tyrosine molecules, and they resemble the steroid hormones by binding to serum proteins when they're acting in the cells. Tryptophan, like I said, after the turkey dinner, gives us that satisfied and tired feeling. And it is also the precursor for our happy hormone or happy neurotransmitter of serotonin, 5-hydroxytryptamine, 5-HT, and also melatonin for our biological clock. All right, so here's a chart that can help you to organize in your mind the different classifications we have of hormones, but there's some overlap, okay? And like I said, you can classify it as non-steroid, but you can get down specifically whether it's a protein or a glycoprotein. And of course, we can classify these things also as whether they're, tropic, whether they're anabolic, if you wanted to do that. Okay, so you can see like growth This thing is going to drive me crazy. I think I'm going to, if I can, switch to my other mouse. Sorry. So you can look here and see like growth hormone classified as a non-steroid proteinaceous hormone, but you could also say it is a anabolic hormone. You could also even say that growth hormone is a tropic hormone. Okay, so when we're looking at our hormones and we're classifying the protein and peptides, you can see here's a long peptide chain. Here it's modified and we get some disulfide bridges. We get further modifications, breaking apart the chains. And you can see there's an alpha chain and a beta chain up here, an alpha chain on insulin with the disulfide bridges formed through cysteines. Steroid hormones are all made from the nucleus of cholesterol. You see this A, B, C, and D ring. And so you see that right here in cholesterol. And so pretty much all of our steroid hormones will have this nuclear portion of it. And then the tyrosine derivatives, this is thyroxine, which is your main thyroid hormone. You can see how it's a derivative of tyrosine here combined. And here's an example of epinephrine. You can see also the nucleus there of the tyrosine. Here again, looking at our hormones, here's the classic example of a proteinaceous hormone of insulin. You can see the chain with the order well characterized of all the amino acids, how they're joined by the disulfide bridges. Here's the alpha chain, here's the beta chain. Here's a much smaller peptide hormone, okay? Only about, 10 amino acids in length. And again, it has some disulfide bridges formed, kind of circulating that from the cysteines. And here's an even smaller hormone, T4, thyroxine, with its two parts of the tyrosine bound to these iodines, okay? Really important, as we know, in our thyroid to have iodine, which is why all our salt is iodized. And then down here, the last one we have is a glycoprotein. The example here is HCG, which if you're pregnant, this is what the pregnancy test measures, beta human chorionic gonadotropin. And you see that you have these long this peptide chain right here, polypeptide, here's another peptide chain, alpha and beta chain, joined with some cysteine residues, but you see it's glycated. It has all these sugar residues on it. All right, so the next group of hormones that we'll look at are all derived from cholesterol. These are going to be our steroid hormones. Okay, the steroid hormones will include hormones like vitamin D. Okay, so vitamin D is a vitamin, but vitamin D is a hormone that travels throughout the body, has many actions. Remember on our bones, for instance. And we have several steroid hormones that are secreted by the adrenal cortex and, of course, the reproductive organs. All adrenal and gonadal steroids have that same basic A, B, C, and D ring structure. Okay, so they have that superficial similarity. It's the side chains that will be modified off of it, which also gives it kind of a different 3D spatial orientation. So it acts with different receptors, very specifically. Anybody asking a question? Yes, I'm sorry, Carrie, that I keep cutting out. My internet is unstable and there's not much I can do about it. All right. So our steroid hormones are all synthesized from cholesterol. And why this is important is you'll recall cholesterol is in every single cell membrane of every cell in your body. Cholesterol is one of the barriers that keeps all those polar substances on either side of the cellular membrane. Lipids are soluble in cholesterol, and so they can pass through the cell membrane and get inside of the target cells, sit on a receptor and have an action inside that cell. So cholesterol, anything that's can get through the cholesterol, like cortisol, estrogen, testosterone, these can travel through the cell membrane and act inside of the cell. Our non-steroid hormones are primarily synthesized from amino acids, proteinaceous, okay, or very small amino acid-like. Now, these are typically going to bind to receptors that are stuck on the outside of the membrane, okay? So kind of they'll come along and they'll stick into a receptor that's poking out from the membrane. But that receptor can have a stem to it that's going to interact with the inside of the cell and trigger what's called a cascade, or one reaction leading to another reaction inside of a cell. Okay, but they don't pass through the membrane. That's the big difference. So here again is cholesterol, our precursor. Okay, we have our 3 6-carbon rings and 1 5-carbon ring. And you see aldosterone, one of our adrenal hormones, looks very much like it. Cortisol, another adrenal cortico hormone, a glucocorticoid, again, looks very much like cholesterol, as does testosterone and estrogen and progesterone. Okay, they all have that same core nucleus. They're all able to pass through the cell membrane. All right. So you don't need to memorize this pathway, but you can see what happens. We have our cholesterol molecule. It gets converted in the mitochondria to pregnenolone. This can go down one pathway, becomes 17-hydroxypregnenolone, which gets converted again to DHEAS, dehydroepiandrosterone. Okay, that's kind of the precursor to a lot of our male hormones. And you can see right down here, converts into androstenedione to testosterone, and also to estradiol 17 beta. I apologize. I'm going to see if I can just hook this other mouse in here and back. Crazy. Put this other one back, get this ancient mouse and see if it works any better and doesn't get so trigger happy. All right, you can see pregnenolone can also go down this other direct pathway and form progesterone, one of our main female hormones, okay? Progesterone can get converted all the way down to our mineralocorticoid, aldosterone. It can stay as progesterone and be active. It can get converted into 17-alpha-hydroxyprogesterone. And this is one of the big things with hormone disorders, is that the endocrinology folks spend a lot of time looking at these pathways and measuring these hormones to see the different enzymes that will convert one of these hormones into another hormone. And you can have a deficiency of an enzyme. Say you could have a deficiency maybe here at this enzyme, and you then insufficiently produce all the hormones that are dependent on that pathway. All right, so this is a lot of what the endocrine testing will do. Say, for instance, there are female hormone disorders where you're born a female, your XX chromosome, but you have deficiencies in being able to stay going down the pathway to making the estrogens. Maybe you have a conversion disorder here at an enzyme. And so you end up with a buildup of a lot of male type hormones, which is a lot of what happens in polycystic ovarian disorder. And so you look very specifically for some of these deficiencies and we test for them. But that gets into pathology beyond the level of this course, okay? All right, so we've looked at amino acid protein type hormones, some cholesterol type hormones. These ones interact, the amino acid and protein ones act on receptors that are in the membrane. Cholesterol, the steroids pass right through the membrane. And then we also have some phospholipid type hormones. These are a confusing array of chemical substances that the major class of which we called eicosanoids, okay? Eicosanoids, it's kind of a big, strange word. And these contain a number of 20 carbon derivatives called prostaglandins, called prostacyclins, thromboxanes, and leukotrienes. And these are also a lot of our inflammatory mitigators. You might remember some of these, like the leukotrienes, okay, will cause a lot of inflammation. So these all are formed from the phospholipids that are in our cell membranes as well. And enzymes can take that arachidonic acid, the basic 20 carbon unit that's in the phospholipid bilayer of our membranes, and these convert into the other chemicals. An enzyme called cyclooxygenase, known as COX, will be active in forming some of these pro-inflammatory substances. So you will see a lot of our drugs are called things like COX inhibitors, like Celebrex, okay? And they stop these arachidonic acid molecules from forming some of these other substances, okay? Usually easier to look at this from a picture, all right? So our eicosanoids, we have our membrane phospholipids, and the enzyme phospholipase A2 will convert these phospholipids into arachidonic acid or arachidonate. Going down one pathway, we have this 5-lipoxygenase enzyme that forms the leukotrienes. Leukotrienes, very pro-inflammatory, okay? And here's different forms of leukotrienes. Down another pathway is cyclooxygenase. This enzyme goes on to form prostaglandin G2, and then we have another cyclooxygenase enzyme that forms prostaglandin H2, which then can split into all these prostaglandins, prostaglandin F, thromboxane. All right, so a lot of these chemicals down here and down here are pro-inflammatory. They cause inflammation. They can cause fever, okay? Prostaglandin E2, well known to help to target at the hypothalamus and cause fever. They can regulate things like, perception of pain, and controlling the thromboxanes control a lot of the regional blood flow. So if we inhibit at cyclooxygenase, we can stop a lot of these molecules that cause inflammation in our body. So these are targets for drugs. Steroid medications like prednisone act up higher on here, stopping things like phospholipase A2. Okay, so it stops both branches, but has greater reaching effects in its side effects. Okay. Okay, Felicia's joining us now. All right, so hormones are going to signal to their target cells by either crossing through the membrane or sitting on a membrane receptor and we say they come in and they sit on their receptor in what's called a lock and key mechanism, much like when we talked about enzymes. They have to fit in really exactly, matching up their chemicals and matching up in a 3D process. Okay, they have to sit in there exactly so that they can trigger the interactions that cause all the chemical reactions, control all the regulatory functions inside the target cell, and thus the target tissues and target organs. Now, every cell is going to have a multitude of different receptors on them, so every cell can react with a lot of different hormones. Okay, when hormones both act in the same way on a cell to have a greater effect, then we call it synergism. All right, business people like to use this term too, of synergies. We have, like-minded business philosophies. Permissiveness is when one hormone lets a second hormone have its full effect on a cell. Okay, permissive. And then of course we have hormones that oppose each other, like glucagon and insulin. Okay, one hormone, insulin, is going to help take all that sugar out of your blood and bring it into the cells that need it for energy. Glucagon does the opposite. It's going to break down your carbohydrate stores to release sugar into your blood when your blood sugar is low. Okay, antagonistic or opposite effects. So here's the lock and key model. All these hormones traveling through the blood. Okay, hormones are released directly into the blood, not through a duct. And then they go to the sites of their target cells. And here, this sort of triangular shape fits directly into the cell, but doesn't fit over in this one that has a circular shape. These are cells that are non-targets. All right, so it only reacts on the cells that have the exact fit receptor for it. When that hormone sits on its receptor, it's going to cause what's known as signal transduction. This is going to unleash, whoops, sorry. This internet is so unstable that I have to keep clicking to try and get this. All right. When this hormone sits on its receptor, see there's part of the receptor that sticks into the cell here, and it's going to unleash a series of chemical reactions that happen in the cell. This is how it regulates all the activities of the cell. And that is called signal transduction, kind of farther down the pathway. Even though it just has this immediate interaction, it leads to 3 or 4 more reactions. Okay, so the signals received by the cell surface receptors are transmitted to the interior of the cell. This could be something like cranking up the formation of a new protein. Maybe it's turning off making proteins. Okay, maybe it's going to open ion channels, so say like calcium can flow in the cell. Maybe it'll open a gate to let insulin inside or glucose inside of a cell. Okay, so the general process of signal transduction is going to let those membrane receptors receive the hormone, take information from the exterior of the cell, and transmit it to the inside of the cell. Inside of the cell, these messages are going to use what we call second messengers that can relay that information throughout the cell. Sometimes it will go on and we will cause a lot of phosphorylation of other proteins to activate them, maybe activate an enzyme. Okay, and so signal transduction and the use of first and second messengers is a little complicated to understand. And God knows, I hope we can meet in person on Monday again so that if you have questions over this, I can go through it with you. All right, on the blackboard. So the primary and secondary actions of hormones are key. Every hormone can have more than one type of action because of all these second messengers. Now, let's look at an example hormone we have here, prolactin. Prolactin regulates lactation, but it has more than 300 other reactions. Most of these other actions will influence, are just influencing the activity of other regulatory mechanisms. Its primary response is lactation, okay? But it can affect other things. Most of our hormones will circulate as either bound to carrier proteins or unbound throughout the blood. So our blood is primarily, the fluid is water, the fluid component. If our hormones are steroid hormones, which are fatty, they're derivatives of cholesterol, you can see that to travel throughout our blood, they kind of need to be wrapped up a little bit or set inside what I always call a protein boat. Because remember, these are going to be hydrophobic. They don't want to be in water. They don't want to be in the solvent of our blood. So being connected to a protein while they travel throughout our body is useful to them, all right? So we have a kind of a little balance or equilibrium of the amount of hormone that's bound to a protein versus what we call the unbound, which is the active form, okay? And it needs to get unbound to get inside the cell and to travel in there and maybe make it to the nucleus of the cell so it can turn on the DNA or whatever it's doing, all right? All right, so we often measure the portion of a hormone that is protein bound and unprotein bound. So when you go and you get your T4 measured, you'll get T4 measured as free T4 and as bound T4. Okay, that's your thyroxine, your thyroid hormone. When the free hormone leaves the circulation to act on its tissue, it becomes freed from that binding protein. Now, we can have specific binding proteins like thyroid binding hormone, but we also have our general multi-purpose protein that kind of sops up and binds to all of our hormones, and that is albumin. Albumin is the major serum protein that will bind to all those lipophilic or hydrophobic, whichever way you want to call it, hormones that need to travel throughout the blood, all right? 95% of our hormones traveling through our circulation are going to be bound, okay? And remember, when they are bound, they aren't free to do their action, okay? And then, of course, they can only act for a certain while. We also have to eliminate them so they aren't always in circulation. And most of these get excreted through our kidneys. Free hormones, or unbound, tend to remain functional in our system for about less than an hour. They'll diffuse out of the bloodstream and act on the receptors in the target cell. And then they're broken down and adsorbed, attached to the cells of the liver or the kidneys, and the breakdown products are excreted through the kidneys into our urine and from the liver into our bile, which ultimately leaves through the feces. And you'll go through that next semester. Or they can be even broken down by enzymes in the plasma or in the interstitial fluids, our tissue fluids. So let's look at thyroid and steroid hormones. These stay in circulation a little bit longer because most of them are bound or attached to a protein. albumin or something like thyroid binding hormone. Okay, so let's talk about albumin. Again, this is what egg whites are, basically, is albumin is the main protein. And this is the protein to which most hormones are bound while it's traveling around in the blood. It's a fatty hormone, okay, a hydrophobic hormone, a lipid-loving hormone, needs that boat in which it's able to circulate throughout the body to get to its target cells. All right. And we call these transport proteins, binding proteins. All steroid hormones are lipid soluble, and so their receptors are normally inside the cytosol, might even be on the nuclear membrane. Okay. Steroid hormones, they make it over to the target cell, they become unbound, and they travel right through the cell membrane. And they're transported into the cell membrane, and then they bind to either receptors in the cytoplasm or maybe on the nuclear membrane, maybe directly in the nucleoplasm itself. And steroid hormones typically will alter the rate of DNA transcription. This changes the way proteins are synthesized. We can have a steroid hormone come in and crank on the DNA and say, keep transcribing this, make some more mRNA so it can travel out into the ribosomes and make more and more of this protein. Okay. An example would be testosterone. Testosterone is going to crank up the DNA transcription of structural proteins like skeletal muscle. This is why testosterone bulks a male up. This is why testosterone can be abused as an anabolic steroid, cranking up that metabolism. All right. You could also have steroid hormones that go in and turn off DNA. Okay. So, and stop the transcription of a protein. We have what's called the mobile receptor model. This is where the hormone will travel into the cell, bind, make it to the nucleus, and then bind to what's called a mobile receptor that activates a specific gene sequence and begin the transcription of that messenger RNA for that sequence. The newly formed messenger RNA, of course, moves out of the nuclear pore and goes over to the ribosomes, either those attached to the endoplasmic reticulum or those free in the cytosol. The steroid hormones, can crank up that regulation and say, let's turn on the manufacturer of a bunch of enzyme proteins, okay, that control a number of other reactions. And so the more you have formed a hormone and receptor complex, the more of this messenger RNA that gets produced, and then the more protein molecules are made for that particular gene. Now, this of course takes a while because you're both telling the DNA to get transcribed more, we're making more RNA, we're having to go out into the cytosol and start, bringing in all the nucleic acids and making all these proteins. This takes a little while to crank up, okay? So the amount of steroid hormone will also determine the magnitude that the cell is going to respond to making that protein. Non-steroid hormones act through that, second messenger system. This is what, instead of calling the mobile receptor model, this is the fixed membrane receptor model. This is where you have a receptor on the outside of the cell, and the non-steroid hormone comes and binds to that receptor that's fixed, sitting right out there on the outside of the cell. It can bind to the outside of the cell. It doesn't directly affect the internal machinery of the cell. Okay, so it's acting on the outside of the cell. But second messengers on the interior, on that kind of stump or part of the receptor that spans through the membrane, will interact with the interior. These are molecules that are going to receive the signal that's on the outside of the cell through that receptor hormone interaction. And it's going to crank up these other enzymes or kind of regulatory proteins in the side of the cell that we call second messengers, like cyclic AMP or G protein coupled receptors. This gets pretty complicated. So important second messengers on the interior of the cell for the non-steroid hormones will be cyclic AMP, which is a derivative of ATP, okay? Cyclic GMP, a derivative of GTP, guanosine triphosphate, or we have a second messenger system through the use of calcium ions. Signal amplification is this thing that I was talking about where we have cascades, where you have one little receptor on the outside of the cell, but it can trigger multitudes of reactions on the inside of the cell through multiple second messengers, all right? It magnifies the effect of one hormone out on the target cell. All right, so G proteins. What are G proteins? They bind to GDP and GTP, which is a lot like ATP, only instead of adenosine, they use guanine. And they will link the hormones to second messengers in enzyme complexes called G protein coupled receptors. We have pictures coming up. They're linked to several different second messenger systems, and they can be either stimulatory or inhibitory. So the G protein can be linked to the second messenger that will either maybe increase cyclic AMP inside of the cell or decrease it. It can also be linked to this calcium system, which is using what's called phospholipase C, okay? That utilize phospholipase C to create a series of reactions that will increase intracellular calcium. G proteins and cyclic AMP will be, as I said, the G protein will activate an enzyme called adenylate cyclase that when it's activated, it will change the concentration of cyclic AMP in the cell. If it increases the level of cyclic AMP, this increases the metabolic activity in a cell. And cyclic AMP moves through a series of steps that first involves activation of protein kinase A. And because it's a kinase are enzymes that will phosphorylate or add a phosphor group to other intracellular proteins. These phosphorylated proteins then undergo a number of physiological actions inside of the cell, and the cyclic AMP then gets degraded to an inactive metabolite. This is what turns off the system. Another second messenger system is G proteins that activate another enzyme called phospholipase C. All right. These activated G proteins will open up calcium channels in the cell membrane, and this will release calcium from intracellular stores, much like in our sarcoplasmic reticulum, right? And these G proteins activate the enzyme phospholipase C. This triggers a cascade of other reactions that produce molecules known as diacylglycerol, DAG, and inositol triphosphate, IP3, to drop out from the membrane phospholipids. These can further activate more calcium ion channels through protein kinase C which then, of course, as a kinase, phosphorylates. Calcium can also activate calmodulin, okay, which causes even more internal cellular changes. Now, this is pretty complicated biochemistry, and I'm not really going to expect you to know this. So you can all kind of go, whoo, but I want you to get a feel for how complicated a single hormone sitting on a receptor can be by unleashing a whole boatload of reactions inside the cell. And this is why you'll see when you get lab tests back, hormones are measured in tiny, tiny amounts, but they have big, big effects because they unleash this whole series of reactions on the interior of the cell, all right? And this is kind of what you need to get the feel for. Forests, rather than all these little trees, all right? This is complicated biochemistry that I don't really expect you to know, but your book goes into it. And so we do really need to kind of talk a little bit about it. And we talked about it, and we're going to talk about it with a picture here. But I'm hoping that you just get the idea more of all these hormones sitting on a receptor causing us big reactions internally in the cell. And there's a high level of regulation, and one little tiny thing at one little enzyme can go wrong and throw the whole system off. And that's why these hormones have such enormous effects in our body. Okay. Now, all right, so let's take a look at this. We'll start out with a hormone right here. Okay. They give the example here of oxytocin. All right. And it comes here, and the hormone is going to bind to a fixed membrane receptor. Okay. So here's the hormone binding to a membrane receptor. All right. And when it binds to this membrane receptor, you have this G protein right here. Okay. And this is connected to an ion channel. So this is a G protein coupled receptor right there. And it's going to activate that ion channel that allows calcium to enter the cell. When calcium enters the cell, that calcium can then bind to this second messenger. Right here is a calcium calmodulin complex. Together, this calcium calmodulin complex on the inside of the cell can then activate or inactivate other enzymes in the cell. You know, if it activates making a substrate into a product, then we'll have lots of this product form. If it inactivates it, we won't have any of the product form, okay? So you'll see that calcium that's normally found out in the extracellular fluid is triggered to enter the cell and diffuse into the cell because of this first messenger binding. But with this second messenger on the inside of the cell, it's going to end up leading to this whole cascade of other events on the interior of the cell. All right. Now, if we look at a typical steroid hormone, here we'll look at thyroid hormone, which can cross. It doesn't have to bind to a receptor out here because it has that cholesterol nucleus to it, right? So it can travel right through the plasma membrane, and it can actually even make it right into the nucleus. When it gets into the nucleus, it can combine with a receptor in here, okay? Now you have the activated hormone receptor complex, which comes over here to the gene on the DNA and turns on the transcription of this gene to form lots of messenger RNA. All that messenger RNA comes back out through the nuclear pore and gets translated into whatever protein we're making out here in the cell. So we have more of this newly formed protein. One hormone molecule going to make boatloads of mRNA, thus leading to boatloads of proteins. Okay, so this is that idea of a cascade, domino effect, right? One thing leads to another, leads to another, leads to another. All right, here's another picture to review a non-steroid hormone, just to get it in our minds. Non-steroid hormone molecule, proteinaceous, right? A protein, maybe just an amino acid. It's hydrophilic. So it doesn't have too much problem traveling around, doesn't need to wrap itself in a protein boat like the steroid hormones. And it doesn't cross through the membrane. It comes and sits on a receptor. That receptor kind of spans here through the membrane, and it's attached to a G protein here. Instead of being attached in the other example, like we had a G protein that was coupled to a calcium ion channel, this one is attached, works with adenylate cyclase. Adenylate cyclase turns ATP into cyclic AMP. Cyclic AMP is a second messenger, okay, in the cell. Cyclic AMP takes inactive protein kinases and turns them into active protein kinases, which tend to phosphorylate a whole bunch of other enzymes in the cell, turning them on. So we can turn on many, many enzymes that might make many, many proteins. This is signal transduction, taking the signal from the outside of the cell and transducing it or transporting it to the inside of the cell. So what's your takeaway here? Your takeaway is that a non-steroid molecule has to attach on the outside of the cell, and you're going to have your hormone receptors going to need to span the membrane and turn on a bunch of second messengers in here. And second messengers will be things like cyclic AMP, kinases, phosphoinositol, okay? All right, I'm not going to make you recall that real specifically. All right, so a nuclear receptor mechanism also exists for non-steroid hormones. Not every single non-steroid hormone operates via second messengers. So we showed you some thyroid hormones before. We can have some small iodinated amino acids, and they can get into the cell and bind to receptors that are already associated with a DNA molecule in the nucleus of the cell. And things like thyroid hormone can affect almost all the cells in the body because it's going to trigger transcription of RNA, right? Similar to what we saw with the steroid hormones. So all of these need to be regulated. So control of making our hormones and secreting our hormones is going to be through endocrine reflexes, largely of which are going to be negative inhibition, negative feedback loops. Remember that from the beginning of the class? Okay, so the simplest thing with a negative feedback loop is going to be where an endocrine gland is sensitive to the hormone that it produces. And when it feels on, the outside of the cell that it's been secreting a lot of that hormone, it feeds back onto the cell itself to turn off the manufacture of the hormone. So we find that the sensitivity of our target cells is dependent on the number of receptors for that hormone. We call it upregulation when we increase the number of hormone receptors, which makes our cells more sensitive to a hormone. Downregulation is when we take all those receptors off the surface of the cell so that we don't have as many of them, thus making our cells a little bit, insensitive to the hormone. We decrease the sensitivity, all right? So let's look at the idea of a feedback loop recall what negative inhibition is, and then we'll look at up and down regulation. All right. So here's a short feedback loop. Our parathyroid hormones, which are little 4 or 5 little pebbles on top of our thyroid glands, all right, always detect, remember, our level of calcium, all right. Each parathyroid gland is sensitive to changes in the hormone that it makes, parathyroid hormone, controlling blood calcium concentration. We also have, when we have lactation, when we're producing milk, a woman needs to consume more calcium, right, to breastfeed the baby. This will, of course, breastfeeding the baby, decrease the amount of calcium we have out in the blood. All right, so we have suckling baby over here, okay? Baby, as we all know, from day number 1, they're little parasites, aren't they? They take everything you'll give them. And so little baby here is getting all the calcium at mom's expense. So mom's calcium decreases out in the blood. All right. This decreased calcium gets detected by our parathyroid glands. All right. Parathyroid senses here decrease calcium. And we know we always have our set point, right? We have our set point where we want it to be, homeostasis. So parathyroids are going to respond by cranking out more parathyroid hormone out here into the blood. Parathyroid hormone is then going to come over to our bones, and it's going to stimulate the osteoclasts to start releasing more calcium from our bone tissue. So the calcium gets released from the bone and out in our blood, increase the calcium level. Okay, this is our feedback loop and makes sense, right? Keeps everything at the nice normal level. All right, let's look at a feedback loop here with insulin production. All right, so we just had Thanksgiving dinner. All right, we ate a whole bunch of turkey, we ate some mashed potatoes, all that, you know, some sweet potatoes, you have your pumpkin pie, your blood glucose starts going up, right? So now we have boatloads of glucose right here out in our blood. Pancreas is going to sense that, says, ooh, I detect high blood glucose. I'm going to crank out some insulin. Insulin works as like the key that unlocks our cells to let the glucose get taken up by all the cells in our body that would like to use the glucose. All right. So it's going to remove the glucose from the blood. Some of it's going to go into the liver and get stored as glycogen. A lot of glucose is hooked together for when we need it in the future. All right. All right. Our blood glucose levels start to go back down. All right. And then maybe we overshoot a little bit and our blood glucose goes a little bit too low. Then our pancreas is going to release the counter-regulatory hormone of glucagon. Glucagon is going to cause that liver that hooked all those glucoses together and stored it as glycogen to say, let's break off some little pieces of glucose and get our blood glucose level back to normal, back to about 100 nanograms per deciliter, okay? All right, and we'll go through this a little bit more and more. We're just introducing right now, okay? The concept of the regulation of the hormones. We'll go through those organs a little bit next time we meet. All right, so we also regulate how sensitive our target cells are to a particular hormone. All right, this is that idea of upregulation and downregulation. So goes to make sense if we put more receptors up out on the cell, right? You're going to catch a ball, right? You're going to catch a hormone out there in the blood. If you only got one receptor out there, the chances that hormone flowing through the blood hits exactly on that receptor is smaller. Okay, so more receptors, greater chance you're going to bind a hormone, greater sensitivity. Hormone receptors are going to be constantly made and stuck on the cell or constantly broken down and internalized. All right, so we're always either upregulating or downregulating, both basically trying to keep, a homeostatic level of receptors. All right, if synthesis is greater than degradation, we have upregulation. If degradation is greater than synthesis, we have downregulation. All right, so here we have upregulation, putting more receptors on the target cell. Here you got a whole lot of them. Downregulation, we start internalizing the receptors, so we won't be as sensitive to all this hormone that's out here in the blood. Now we have some control sources. Well, like I said, the main method is usually always negative feedback for everything in the body. There are a few instances, mainly in the reproductive system with oxytocin, having a baby. And as we said before, the other thing with positive feedback tends to be our clotting system. All right. So we'll talk a little bit more about negative feedback because that's our main method that we use. All right. So 3 methods in which negative feedback is going to control the secretion of hormones in our system will be first, number 1, tropic hormones. Tropic hormones are hormones that will act on other glands in order to regulate their secretion of yet another hormone. Yes, I'm in here. Okay. The other route is through the nervous system control. Here, the nervous system can stimulate some glands to secrete their hormones. So a nervous impulse comes into the gland, and the gland, like our adrenal medulla, is stimulated, as we learned through the ANS, right, to produce some norepinephrine, which can then travel throughout our circulation and go to, say, our heart. The third method is that we get changes in the composition of our internal environment. So you might get changing levels of a specific substance out in the blood. We just went through glucose, right? That if we have high glucose levels, it's going to stimulate the secretion of insulin, so our cells take up the glucose. Our liver cells, for instance, will take up the glucose and knit them together to make glycogen. So we can use it later on when we need it. Or we can inhibit the secretion of another hormone, okay? So we actually inhibit, in that sense, the secretion of glucagon at that time because we don't want to break down glycogen. We already got a lot of glucogen. We already have a lot of glucose out in our blood. All right, so let's look at some of these feedback mechanisms and how it works. Remember how I said you have kind of 2 master glands? So call the pituitary gland the master gland, and we'll call the hypothalamus the grand master because it controls the pituitary. All right, so we can have a tropic hormone. In this instance, our hypothalamus will stimulate our anterior pituitary, or anterior hypophysis, to secrete a hormone that acts out on a peripheral gland like our thyroid or our adrenal gland. This will then in turn stimulate our thyroid gland, or like our adrenal gland, let's say our thyroid gland, to secrete its hormone, thyroxine, T4 or T3, okay? That hormone then goes on to act on the target cells, all the cells in your body that need to have its metabolism stimulated by the thyroid hormone. And that's ultimately the action, okay? You can also have part of the brain, the nervous system, stimulate an endocrine gland. We'll see how we also have what's called like the pars nervosa part of our pituitary gland that's stimulated by the nervous system, which can then go on to affect some other target cells to then have their action. Now, what we see out here is when we have that action building up, maybe the release of a bunch of hormone out into our system, that hormone builds up and it feeds back on the nervous system. This is a negative feedback loop and turns it off because this has had enough action. Here, we could see where maybe if we had like our thyroid gland, okay, is stimulated to produce thyroid hormone to act on target cells. If we get a lot of T4 and T3 out there, it's going to feed back at one level on the pituitary gland and say, oh, stop sending out thyroid stimulating hormone to stimulate the thyroid gland, okay? Or it can also feed back into the brain on the hypothalamus and say, stop sending out thyroid releasing hormone. Thyroid releasing hormone tells the anterior pituitary to send out thyroid stimulating hormone that then tells the thyroid gland to start sending out thyroid hormone, which is sent out to all the cells in the body to increase their metabolism. All right. Now, changing the level of substance in a plasma is like glucose. We have let's say we have a change in glucose where our glucose goes down, okay? We have low glucose out here. This is going to incite our pancreas to not release insulin, okay? Okay, because insulin is going to go out to our target cells and open them up to start clearing glucose out of our blood, but we don't have enough glucose. So it turns it off and feeds back out here until we build up the level of glucose. It's also going to affect the endocrine gland of the pancreas to crank out glucagon, okay? Glucagon is going to cause the liver to start breaking down glycogen and release lots of glucose out into the plasma, also building up the substance of glucose out in the plasma. All right, I know that's a lot, but we have, a little bit of time yet to discuss our hormones. So don't freak out too much yet. So hormone regulation and secretion, definitely mostly controlled by negative feedback mechanisms. Negative feedback is when the substance itself is going to build up and turn off the process that produces it, right? Okay, the stimulus triggers the production of the hormone, and the direct or indirect effects of that hormone will reduce the intensity of the stimulus. Now remember, hormones effects can be either short-lived or act over days, weeks, maybe even a month, all right? Hormone secretions have to be very precisely regulated, and most of our hormones are excreted from the kidneys after they exert their effects. We pee it out in our urine. Hormones can also be broken down by some enzymes, mainly in the liver, to stop their effects. Okay, so the control of hormone release. Whoops. Let's look at the level, like I said, at the hypothalamus, our grandmaster gland. Our hypothalamus is largely going to produce what we call both releasing hormones, stimulating a gland like our pituitary to start the flow of release of hormones, or some inhibiting hormones to stop that, all right? The pituitary level then produces stimulating hormones like thyroid stimulating hormone. This is going to stimulate the end organ or endocrine gland to produce its effector hormone, like thyroxine, T3 and T4. Okay, so hypothalamus, example, thyroid releasing hormone. This prompts the anterior pituitary to release thyroid stimulating hormone. Thyroid stimulating hormone then goes to the thyroid gland and causes it to release thyroxine, which comes in two forms. T3, a highly active form, which we secrete in a low amount, and T4, the more common form, a little bit less active. All right. When you build up all this thyroxine out in your blood system, your thyroid's been producing a lot of it, it will feed back on both the anterior pituitary, turning off thyroid stimulating hormone, and it will also feed back on thyroid hormone, also turning that off a little bit to control it to not stimulate the anterior pituitary. Okay, so if we looked at this and we wanted to draw it in here, we could take our little pen here and say this is going to feed back here, okay, like this, and it can turn this off when this goes up out in the blood, right? It can also negatively feed back on this, stop producing that, okay? All right. Now, let's look at eicosanoids. Eicosanoids, big word, always kind of hard to understand. This is that, these are a group of hormones that form from arachidonic acid, which rests in the plasma membrane of cells. Okay, so we have steroid hormones that are made from cholesterol and can travel through cell membranes because cholesterol is one of the fatty components of all of our cell membranes. We also have arachidonic acid, which is one of those fatty acid kind of things, tails that hangs off in our cell membranes, 20 carbon fatty acid, all right? And this can have enzymes act upon the arachidonic acid. We already looked at cyclooxygenase and lipoxygenase to form all these sort of tissue hormones and pro-inflammatory substances. Tissue hormones are secreted in tissues and they diffuse only a short distance in that same tissue and are rapidly metabolized, okay? Prostaglandins are named because they were first described in the prostate gland, but we now realize, of course, that these are made pretty much everywhere throughout the body. These are what we call lipid autocoids, okay, derived from that arachidonic acid in the cell membrane, and their biosynthesis of these prostaglandins are formed by those enzymes lipoxygenase and cyclooxygenase. And of course, the drugs that we take to inhibit those enzymes stop these compounds, these pro-inflammatory compounds, from being made. We have specific COX inhibitors like Celebrex, okay, or non-steroidal anti-inflammatory drugs. They both stop this process. They stop these substances from being formed from arachidonic acid. All right, let's look at that again. So you have this 20 carbon phospholipid. Phospholipase A2 is the enzyme that allows arachidonic acid to be formed. At this level, this is where glucocorticoids, prednisone, works to stop this process so you don't make either branch, okay? From arachidonic acid, we have COX, the cyclooxygenase enzyme that forms these prostaglandins and thromboxanes. NSAIDs, okay, non-steroidal anti-inflammatory drugs, things like your Aleve, your ibuprofen, things like Celebrex, which is a little bit more specific, okay, COX inhibitor. these all act over here, all right? The lipoxygenase process allows leukotrienes to form, okay? So you can see how glucocorticoids inhibit both of these. And as you know, taking a steroid like prednisone is a little bit more powerful anti-inflammatory because it controls both pathways than is ibuprofen, okay, which controls one pathway. Of course, as you also know, taking prednisone will have then more side effects than will taking Aleve or ibuprofen, okay, because it's happening lower down in the cascade, lower down in the chain of reactions. I hope that makes a little bit of sense to you there. And that's really what I'm trying to get the picture for you is, you know, the higher up in a chain of reactions that you're going to get a drug to block things here. And this is why we want to learn this. You want to know what all these drugs, I mean, prednisone, you're going to be seeing patients. Most of you have probably taken it. I'm sure Kiaria has taken it probably pretty recently. All right. The glucocorticoids act up a lot higher, have a more profound effect higher up in a series of reactions than something that acts much more specifically lower down in a series of reactions. Eicosanases, as I said, are what we call tissue hormones because it's produced actually in that tissue right there, and they only have to act a short distance on the cells right next door to each other in that same tissue, okay? Prostaglandins, many tissues, like I said, even though they're named prostaglandins from the prostate, it's made in so many tissues throughout our body, and they have diverse physiological effects, which really starts getting beyond the scope of our class, okay? They are paracrine substances, and they're potent in small amounts like hormones, and they're not stored in cells. They're just synthesized right before they're released and then used and rapidly inactivated. And they can either activate or inhibit adenylate cyclase depending on the particular tissue. Like I said, this gets a little bit beyond the scope of our class, but you'll see at different times that, you know, prostaglandins might either relax or cause smooth muscle to relax or contract. They can help regulate your blood pressure by acting on the vascular smooth muscle. So you will see that they have pretty diverse range of effects throughout the body. Now, most of our endocrine disorders typically result from us either having too much hormone or too little hormone. When we have too much hormone production, we usually call this a hyper situation, like hyperthyroidism, too much thyroid hormone. Often this is caused by having an abnormal proliferation, too many endocrine cells making their product. Okay, so you might have a tumor, maybe you just have hyperplasia, just like, you know, an excess of cells there, but they're not actually a tumor. You can have an autoimmune disorder, okay? Autoimmune disorder might have something where you're attacking your own your own cells, and maybe it causes your feedback system to turn off. So you just keep producing hormones without having any regulation. So let's have an example. Hyperthyroidism. It's not really a disease, but it's many different kind of diseases can cause hyperthyroidism. So for instance, we can have the autoimmune disorder, Graves' disease, okay? And in this case, you produce immune system cells against your thyroid disease and you lose that feedback mechanism. And you produce too much thyroid hormone. And then what happens? You get the jitters, you start losing weight, your eyes start bulging out, okay? You get really thin, you get really fast, you have wasting, you get high blood pressure, your heart palpitates. And we'll see hypothyroidism symptoms are the opposite, okay? You could have a thyroid tumor that also did the same thing, okay? And that's not Graves' disease. Now, you can also have, as I said, hyposecretion, okay? Decreased levels of the hormones. You could have this caused by a tumor, okay? That maybe you have a different cell type that replaces the normal cells there and you stop producing the hormone. You could have tissue death. Maybe you had the blood supply to your thyroid gland being blocked. You know, you had a clot there or something and the tissue dies off. And so you stop producing the hormone. Maybe you have an autoimmune destruction of the cells. This is what causes type 1 or juvenile diabetes. You have an autoimmune system disorder that destroys your pancreatic islet cells, so you stop making insulin altogether. That's why you can't treat type 1 diabetes the same way as type 2. You have to give type 1 diabetics insulin because they just can't make it themselves. All right. Or people often do stupid things. Like they think I'm going to build up a whole bunch of muscle by giving myself some anabolic steroids. I'm going to take a lot of extra testosterone. You know, a little bit of testosterone makes me manly. An extra dose of testosterone is going to make me super manly. Well, what happens when men take testosterone enhancing steroids is that they increase their blood level of testosterone above their set point, and the feedback loop then causes them to stop producing testosterone themselves in their own body. Okay. So you're doing yourself a disservice. All right. And then you have to keep giving yourself more and more exogenous testosterone. All right. So we've done enough for today, probably confusing ourselves a little bit, but we'll wind back on Monday and we'll ask a lot of questions and we'll be back in class for sure by then. I just can't imagine that my test results cannot be back by then, and I'm feeling fine. I got a second test today or yesterday just to ensure that we get results. All right, so in comparison with the nervous system, the regulatory effects of the endocrine system are going to be more slow to appear, but longer lasting. The endocrine glands themselves are made up of which? Glandular epithelium, neurosecretory tissue, ducts leading to major arteries, or both A and B. I think from the types of answers you can tell it's going to be both A and B. No ducts in the endocrine system, okay, ever. Endocrine system dumps everything directly into the blood, okay? Good job, Maya. All right. Let's get on to this next one. Sorry, internet's unstable again. All right, all of the following are non-steroid hormones. This is a pure memorization question. So this is, you know, can you remember from earlier when we said steroid hormones are cortisol, estrogen, progesterone, testosterone, all those things that have a cholesterol nucleus, all right? And you'll get more and more of a feel for this as you read through the text and go through the homework questions, or I guess PBL questions, and, you know, as you advance in your career, okay? Cortisol, one of the adrenal hormones. Steroids are able to pass through a target cell's plasma membrane because they are
lipid soluble.
Exactly. That was Kyria, wasn't it? I recognize your voice. Lady who's already done. All right. They're lipid soluble. All right. They need a boat of protein, a binding hormone, right, to travel around in the circulation. But once they get to the cell, they actually just kind of meld right in through the cell membrane. Whereas the non-steroid hormones have no problem traveling throughout the circulation, but they can't get into the cell easily. So they tend to bind out on external cell receptors. Okay. Downregulation refers to which one of these? We're going to take receptors, right? And we're going to internalize them back in the cell. So we're going to reduce the number of hormone receptors. If we upregulate hormone receptors, we're going to stick more and more of them out to increase the sensitivity so we can capture a hormone, more likely to do so. Which of the following is not a protein hormone? Okay, so before we were saying what are the steroid hormones? So this is the opposite. So protein hormones, oxytocin, that's a small amino acid. Insulin,
Yeah,
that's also proteins, right? With the cysteine sulfide bonds. Parathyroid hormone. What one is this? Does that look like a steroid? Or is that more of a small kind of amino acid?
amino acid.
There you go. And Regina raised your hand. Regina got a question? Or is she going to answer it? Okay. Now, I take difference with this question a little bit, and I would probably give you the credit on this question because oxytocin is not legitimately classified as a whole protein. It's a small amino acid, like I said, but you know, I really think that's splitting hairs. So I kind of would give you credit for saying all the above are protein hormones. All right. All right, the lock and key mechanism allows hormones to bind only with the target cells that have receptors that fit with them exactly. True, right?
True.
This is true. This is
Yes.
just like an enzyme too. Enzymes and hormones, highly specific to the chemicals fitting in three-dimensionally. Endocrine glands release their hormones into ducts that eventually empty into the circulatory system.
False.
False. That word ducts, right? Ducts.
Yeah.
Released directly into the blood. No duct involved. That's the difference of exocrine and endocrine. Exocrine released into ducts that goes out on the epithelium. Yep, Regina? Okay. Yep. Regina, can you type a message in the chat? I can't hear you. All right, and I think we're done. Oh, we're done. All right, so I wanted to give you this extra time so you have time to work on your exams or get some extra sleep so that you can get up early, you know, and spend some time tomorrow or that. And apparently Kyria doesn't need it.
Oh my goodness.
I can't get over that. I've never had anybody finish class early like that. So that's pretty amazing. That's really using your recovery time wisely. Anybody have any questions? Kind of work with this material and, you know, we'll go through more specifically some of the hormone systems on Monday. And if you have questions about negative feedback, we'll also go over more of the hormone disorders like diabetes and that, because obviously it is the plague of America. And I hope you're all doing well. I hope nobody knew anyone out there in Oxford area tonight. I heard there was a 4th death and, you know, I hope for better for us for the holidays, don't y'all? So, so I will see you Monday. Everybody stay happy and healthy and do well. Okay. Send me your questions if you have any questions. Okay.
Okay. Thank you.
Yep. Bye-bye.
Bye-bye.